What is tetanus? Tetanus is a disease of the nervous system caused by spores of the bacterium Clostridium tetani, which is most commonly found in the soil and in animal feces and mouth. Patients contract the infection when open wounds are contaminated with the spores. When the spores enter the patient's wound, they become active bacteria that spread throughout the body, releasing the poisonous neurotoxin tetanospasmin. This toxin causes muscle spasms in the jaw, chest, back, neck, and abdominal muscles. As a result, this can lead to things like airway obstruction, respiratory arrest, heart failure, damage to muscles, fractures, brain damage, and eventually death. How does one toxin cause so much damage? To answer that question, we need to travel to the spine, where the signal transduction pathway of the lower motor neurons, which are the nerve cells that activate voluntary muscle, are located. In this pathway, there are two types of neurons that connect to the motor neurons that connect to the muscle. The first neuron that connects to the motor neuron is the inhibitory neuron. When it receives signals from the brain, voltage-gated channels open, triggering an influx of calcium ions in the presynaptic membrane. With the help of snare proteins, vesicles holding GABA, the inhibitory neurotransmitter, fuse with the presynaptic membrane. Once GABA is released into the synapse, it travels to bind with the ligand-gated channels on the postsynaptic membrane, which is in this case a part of the motor neuron. Binding with the ligand-gated channels allows for the flow of chlorine ions into the motor neuron and the release of potassium ions. This leads to the postsynaptic membrane to hyperpolarize, or become more polar as the flow of ions goes against the concentration gradient. Polarization causes inhibitory postsynaptic potential in the motor neuron. Later, I'll explain how important inhibitory potential really is. Before that, though, I have to talk about excitatory potential and what it has to do with muscle contraction. Excitatory potential is generated in the excitatory neuron that I showed earlier. It is generated in a similar fashion to the inhibitory potential, but instead of the vesicles releasing GABA, they release acetylcholine, or ACH for short. When ACH binds the ligand-gated ion channels of the postsynaptic membrane, it allows for sodium ions to flow into the motor neuron and potassium ions to flow out. This causes depolarization, the opposite of hyperpolarization. This means the flow of ions goes with the natural concentration gradient of the membrane, making it less polar. Depolarization of the postsynaptic membrane is what causes excitatory potential, as it releases stored potential energy. Excitatory potential transfers action potential to the postsynaptic cell, which is in this case the motor neuron. From there, a similar process ensues. As the excitatory postsynaptic potential reaches the motor neuron through the same process as the excitatory neuron, it releases ACH neurotransmitters into the synapse. This synapse, however, is called the neuromuscular junction, as it is here that the action potential from the motor neuron is transferred to the muscle. The released ACH travels across the neuromuscular joint towards the motor end plate and binds with the nicotinic ACH receptors, triggering ACH-gated ion channels to open. This allows an influx of sodium ions into the muscle membrane, creating end plate potential and generating and transmitting action potential to the muscle membrane. This action potential then leads to muscle contraction. So, going back, inhibitory potential is basically the opposite of excitatory potential. Where excitatory potential depolarizes, inhibitory potential polarizes, controlling the excitatory potential and modulating the force of muscle contraction. Now, you may be wondering, how does tetanus affect this flow of messages within the nervous system? First, it is taken up in receptors of lower motor neurons. It travels retrograde, or backwards, through the axons to the spinal cord and brainstem. From there, the toxin moves into the inhibitory neurons that, like I mentioned earlier, control the activity of the lower motor neurons. In the inhibitory neurons, the toxin cleaves the VAMP protein, which is a part of the snare protein complex that I mentioned earlier. VAMP, as a part of the snare complex, is necessary for the release of GABA through fusion of synaptic vesicles with the neuronal plasma membrane. In this way, vesicular release of inhibitory neurotransmitters becomes blocked, leading to disinhibition of lower motor neurons. This creates hyperactivity in the motor neuron, and as there is no inhibitory neurotransmitter, leading to the muscle spasms and rigidity that characterize the tetanus disease.